Hi, and welcome to the final video on macromolecules. So we're going to talk about proteins, and then that's going to be it. So our elements that make up proteins, we have carbon, hydrogen, oxygen, sulfur, and nitrogen. So these five elements come together to make proteins. So we have carbohydrates with carbon, hydrogen, oxygen, lipids with the same exact stuff, carbon, hydrogen, oxygen, and sometimes phosphorus. Nitrogen has carbon, hydrogen, oxygen, nitrogen, and phosphorus. And then now for proteins, we have carbon, hydrogen, oxygen, sulfur, nitrogen. So I like to think about cho, chomp, and then chosen. Cho, chomp, chosen. So we've got carbs, lipids, proteins, nucleic acids. Proteins are chosen. Nucleic acids have chomp. So monomer for proteins. Once again, we're looking at the individual little bits for proteins. So I've got my little chain here. This is going to be a monomer. It is an individual unit. And then when we link it together, it creates something known as a polypeptide chain. And that polypeptide chain will eventually be folded until it becomes a functional molecule. Of course, this isn't functional, it doesn't work right now, but it would eventually become a protein. Now, these amino acids, the monomers here, are held together by something called a peptide bond. Peptide bond. So we have a polypeptide chain with peptide bonds in between the amino acids. So our amino acid structure. The only reason why I'm going to go over this is to show you guys that scientists aren't crazy by naming it this. They do have some logic in the name. So we have carbon in the middle. Once again, it has 1, 2, 3, 4 different bonds that it's going to be able to make here. The top one's going to be hydrogen. That one really doesn't do too much. We're not worried about it. Over on the left-hand side, we have a nitrogen group, and this nitrogen group, NH3, is known as ammonia. Ammonia. We have some similarities here. Ammonia, amino. And then over on the right-hand side, we have COOH. And you might remember that acids are characterized by dissociating hydrogen ions. We have a lot of hydrogen ions there. Well, this guy over here really, really loves to get rid of his hydrogen molecule, which means that it is an acid. So in this entire molecule, we have amino, which is ammonia. We have an acid structure. And then this R group just stands for something that will change. Kind of like X in algebra and how X is a variable. It can be just about any number. R here is going to be just about any molecule that we can stick onto that amino acid structure. So the R group. Over here, and I flipped the molecule upside down. I'm sorry about that. We've got the acid over here, we've got the amino group over here, and then up on top you can see how this is a CH2 with like a little ring, and then up here it's just CH3. So that R group will change just like how in math you have a X and a variable. So these are two different amino acids. The top one is called alanine and the bottom one is phenylalanine. And they will link together in order to make your proteins. Amino acids link together to create the protein. That's on your test. So you can see how the amino groups and the acid like to bond with each other. They do a really, really good job sharing that. And that bond is called the peptide bond. Now, uses for proteins. It's like everything in your body. It's structural, forms your bones, forms your muscles, forms your skin. Your hair, skin, and nails, the top layer is called keratin, and that keratin is a really, really wonderful protein that helps keep everything together and makes your skin tough and waterproof, just like how your hair and nails are pretty tough and waterproof as well. So enzymes are another type of protein. And I mentioned one earlier when you will eat something and it'll dissolve in your mouth. Enzymes are proteins that will make a reaction happen very, very fast. So enzymes speed up the rate of reactions. They speed up the rate of reactions. Molecules will break down on their own, but it can take a really, really long time, and we don't have enough time to wait for all of that to happen. So instead, we have enzymes to speed up that reaction. And then we also have proteins that allow for transferring things, bringing things into and out of our cells. That way we have enough stuff to actually function. Antibodies, these are little proteins that will attach themselves to things that need to be destroyed by your body. So if you are really, really allergic to pollen, what will end up happening is that antibodies will attach themselves to the pollen and signal to your immune system to attack and kill that pollen. Of course, pollen isn't actually bad for you. It's just a problem that your immune system has with misidentifying a painful or harmful thing. But we end up seeing antibodies work really well against bacteria, and antibodies will also even mark like cancerous cells to destroy those as well. Antibodies will help keep your body safe. So let's go back to the enzymes. Enzymes are known as a catalyst for the body. A catalyst for the body. A catalyst will speed up the rate of a reaction, and enzymes are just molecules that speed up the rate of reactions in your body, and they happen to be proteins at the same time. So, catalyst speeds up the rate of a reaction, and on your little station things, we had things called substrates and enzymes. The substrate is another word for the reactant. I'm sorry that we have multiple names for the same exact thing, but here we are, and you gotta know it. So, a substrate, otherwise known as a reactant, enters the active site of an enzyme in order to form the enzyme-substrate complex. So essentially, the thing that is going to be broken down ends up attaching itself to the enzyme, and this is what makes the enzyme-substrate complex. After it, let's say that it breaks apart, talking with my hands now. It's going to leave the reaction, but the enzyme is still there and intact in order to do that reaction all over again. So after the reaction, the product leaves. The enzyme remains unchanged. It's still there. And so this is an example of the enzyme substrate complex and then it leaving. Instead of this being an example of us breaking apart a molecule, this is an example of us forming a larger molecule. We're going to see this a lot when your muscles are trying to get bigger. Enzymes are there in order to build bigger proteins and make this stuff come together. So we're going to see this especially in cases of dehydration synthesis when we're making longer molecules like storing sugar in your liver or building muscles or bone or something else. So enzymes. Types of protein. Oh my gosh, please remember this. Enzymes are a type of protein, just like how Nike is a type of shoe. Within enzymes, we've got the enzyme substrate complex. These are the substrates, the reactants. We've got the enzyme, and then the product leaves. Don't forget about reactants versus products. So the enzyme speeds up the rate of reaction. And it almost works like a lock and key. And this is something that you will see not only in this biology class, but in just about every other biology class that you take. So, an analogy for you. I need something to happen quickly. Let's say I need to open the door quickly. The door is going to be the reaction. I need to separate the actual door from its frame. Now, if I were to leave school and find a magic potion that lets me live for thousands of years, I could come back to the school after 2,000 years and I'm sure that that door will be knocked down. But I don't have time to wait for that, and chances are I'm not going to find a potion like that. So instead, what I can do is use a key in order to separate the door from the door frame, and that will allow me into the room. The key is specific to my door. That door can only get unlocked by this key. I can't use this key to unlock Mr. Nelson's door. I can't even use this key to get into the gym. I can only use this key on my door. So this key, that door, this is going to be the enzyme. The door and the frame is going to be the substrate. Now, along with that, I don't have to get a new key every time I unlock my door. That would be very inconvenient. Instead, this enzyme is constantly being able to use. I'm always able to use this key for that door, this key for that door. So we've got an enzyme as a lock and a key. Energy reaction diagrams. So when we look at the science, at the numbers, at the energy level, how do enzymes work? Well, they just make it easier for the reaction to happen. If you think back to energy absorbing and energy releasing, Hopefully we got this. 1 and 2, energy absorbing is going to be diagram 1. Energy releasing is going to be diagram 2. Enzymes change the reactions by lowering the activation energy. So this is an energy releasing reaction. You can tell because it goes up and then it falls down. There's more energy in the reactants than there is in the products. So because this goes up and goes down, there's a lot of energy needed in order to get this reaction started. Going back to the campfire analogy that we had earlier in the day, it's going to take a lot of work in order for me to get a campfire up and running. I have to collect all the sticks. I have to get the heat generated in order to produce the fire. An enzyme would be like throwing gasoline in a match on the campfire. It's going to be a lot less work, and that fire is going to go for quite a bit of time. So we end up having enzymes lower the activation energy, lower the amount of energy needed to put into the reaction. This is like, let's say you get a new phone, your dad is amazing, and your dad ends up like activating your phone for you so that way you can use it right away. So lowers the activation energy. Now, enzyme reaction rates. There are a lot of different things that can change how your enzymes work, just like how there's a lot of different ways that could change my key from working. These rates can be affected by a lot of different stuff. First thing is temperature. If it is really, really, really hot, this key, made of metal, will melt. A melted key is going to do me no good. Now, thankfully, we don't have to worry about that too much, but metal will melt and your enzymes will definitely melt as well. Now, your enzymes are much more sensitive to temperature change. They work best at 98.6 degrees Fahrenheit. Hopefully that sounds a little familiar to you guys. 98.6 degrees Fahrenheit is the average body temperature of a healthy human. So enzymes work best at that temperature. Any lower and the enzymes will tend to slow down. And any hotter, especially at 113 degrees Fahrenheit, the enzymes will literally melt. And at 113 degrees Fahrenheit, you can die because those enzymes have all melted, which means that you can't get any fast reactions done in your body. pH, acids and bases can alter it, and how many enzymes you have. If I have one key and I lose it, I'm going to be out of luck. Hopefully I find it soon enough to open up the door. However, if I've made tons of copies of that key, it's going to be a lot easier for me to get into and out of my room. And that is it. You guys need to have your summaries for both carbohydrates, lipids, proteins, nucleic acids, and I guess all the monomers and polymers and all that stuff. So I guess not both, but all of them. We are going to have a test here pretty soon. So make sure that you guys do have this filled out. And if you have any questions, please, please, please feel free to ask me. Thank you for watching.